So let's start. Dear participants, ladies and gentlemen from all over the world, as the president of Iabs, it's a real pleasure for me to welcome you at this webinar we organized together with CEPI. New platform technologies such as messenger RNA, viral particles, recombinant construct highly contributed to a quick response to the COVID-19 pandemic. In an unprecedented speed, vaccines have become available within less than 1 year, and also on the treatment, on the antibody side, a huge progress is possible thanks to platform technologies. All the speakers and panel participants, which are warmly welcome, and I thank really for their involvement and participation because they all have impossible agendas. They all will develop one or more aspects of platform technologies, and particularly the regulatory aspect. With CEPI, we believe it is a good moment to exchange now on platform technologies in order to get more out of it for this and let's hope not, but it's almost sure, other pandemics to come. I wish you a very good and exciting webinar, and I hand over now to Professor Peter Niels for practical information and leading this webinar. Peter.
Thank you, Joris. I also want to welcome everybody to this webinar. I'm Peter Niels. I am the chair of the Human Vaccine Committee at Iabs, and we organize this, as said, together with CEPI. The first announcement I have to do is a little bit disturbing. We learned this week that Mark van Ooy from Johnson, who is on the agenda, is not able to attend, and so we have to skip his presentation. But I hope that we will be able to have the other presentations very quickly. And I hand over to Melanie Seville from CEPI. Melanie, the floor is yours.
Thank you very much, Peter and Joris, and just to add my welcome from CEPI to this webinar. Platform technologies, in particular rapid response platform technologies, have been something really at the heart of what CEPI has always been doing. So looking at initially, obviously, a situation of disease X and how vaccines can be more rapidly developed through often the more modern technologies. And we have all seen with COVID-19, our disease X coming, and seen tremendous progress being made in terms of the speed of development of vaccines against COVID-19. And this is clearly something to be built on. And we're already seeing with COVID-19 the need for continued to develop platform technologies to speed up development with the emergence of new variants. And many of the developers are already working on new variants, but as already pointed out, looking for being better prepared for future pandemics that we know will emerge. So it is a really good opportunity at this point in time to take a step back, to get some experience from industry, some views also on all of the work that's being done in the veterinary field through ZAPI, that I think from a human perspective we can all learn from, as well as having the regulators' perspectives on the topic. So really, without further ado, I think we should get into the meat of the discussion. And Joris, I think I hand back to you for your presentation on ZAPI. Thank you.
Thanks a lot, Melanie, for this beautiful introduction. I share my screen. Do you all see the presentation?
Yes, we do.
Okay.
Yes, we do.
So it's a pleasure for me to talk about the Zoonosis Anticipation and Preparedness Initiative called ZAPI, which is one of the multiple IMI Innovative Medicines Initiatives, a public-private partnership of the European Union. And the ZAPI initiative was started and at the end of 2014. And when we first discussed that as a preparation, late 2014 and early 2015, we hoped that we would not be as actual as we are, unfortunately, today with the pandemic. So ZAPI itself, it's a One Health initiative where we try to use the best of both human and veterinary medicines and experiences. And the objective was in order to be better prepared for pandemics, panzootics, by developing only one, and there are many, as you know, platform technology, which we call multimeric protein scaffold particles. So it's a scaffold where you glue in antigens and then you have a vaccine. Very simple to explain. And we used for that 3 model viruses. MERS coronavirus, which most of you know probably, unfortunately I would say. Rift Valley, who is a zoonotic virus, and also Schmallenberg, who is also a Bouwenia virus like Rift Valley, with a potential to become zoonotic. The objective was to try to investigate and doing research on vaccines and antibodies by design in a flexible way to generate robustness of the platform and prepare for surge capacity in case of emergencies. Vaccine prototypes have been developed for all 3 model viruses, and antibody platforms have been developed for MERS and Rift. And in ZAPI, we go till the pilot project just before the clinical phase. A very important part of the ZAPI project was regulatory innovation, how to see we can include this innovation in the regulatory science and regulatory pathways. And for that, we drafted, and I like to thank one member particularly of ZAPI is Dr. Carmen Jungbeck, who worked a lot on the platform master file. What we mean by platform master file is a part of a licensing dossier intended to fasten up the regulatory process, keeping in mind that we cannot compromise on safety and efficacy. And it has another objective, avoiding to repeat discussions of already accepted platforms and particularly data on platforms. So the goal is to enable a swift response to new infectious disease threats. And in ZAPI, we have 2 main parts. The first, the scientific part. And the scientific part is very comprehensive. It starts from a methodology in silico, for example, where you try to find very quickly the genome and the important epitopes and the important characteristics of a pathogen in order to design a vaccine antibody and be able to react quickly. So in order to prepare for surge production for vaccines, therapeutics, and in particular viruses. The regulatory aspect is the consequence of that, and during the whole process, the regulatory activities accompanied the science and then the development in order to be able to propose to regulatory authorities and exchange with them. And by the way, we had many contacts with European regulatory authorities, and I like to thank them at this occasion for the very open and interesting exchanges we had. So also propose recommendations for approval and how we can best use platform technologies in emergency situation. And at the end, I will come back on the example of COVID, of course. So the platform master file, it's a standard, we consider it as a standalone part of a marketing authorization. And by platform itself, we mean the backbone, and the backbone can be a vector, or it can be, for example, a virus-like particle, like we developed the multimeric protein scaffold particle. It can be messenger RNA, it can be DNA, it might be replicants, viral and bacterial vectors. And it also concerned the expression of antigens where needed for subunit vaccines or viral particle vaccines, for example, new expression systems like fungi. The relevant information, as I said, does not and may not compromise on quality, safety, and efficacy, but it can be seen in an emergency status, as we have today. It's a standalone part, and it can be common for one or more immunological medicinal platforms or products. Of course, when you have your pilot, you need to prove safety and efficacy, which has been magnificently done for some COVID vaccines already today. The situation to date, from a legal aspect, so we worked particularly on the veterinary side for the vaccines, and we had a lot of exchanges in the writing and with the writing on the new veterinary medicinal regulation in the EU. And it was the opportunity also to draft this new kind of technology technologies, the platform technologies, in order to include it in a regulatory approach. Now we are so far that the whole regulation follows the legal path of the consultation procedure, and legally it will be in place as from the 1st of January 2022. So it includes further safety and clinical data for veterinary and human medicines. This inclusion will be part of, must be part of clinical trials. A very important thing with the experience we have and the experience that we learned already in the recent year, would it be possible? That's a question we pose. And would it be an opportunity for shortening or simplifying phase 3 trials based on all the data generated? And what can we learn in this respect, for example, from the COVID-19 messenger RNA vaccines? Not to forget, and Melanie already alluded to that, can the platform master file principle be useful for adapting existing vaccines to variants? And in the face of a pandemic, can we speed up the use of new technologies, of course, subject that they prove safety and efficacy, like we have seen with the messenger RNA, and like we could see with alternative expression systems like fungi. The content in brief, as we proposed it, is a detailed description of the expression system and the construct, of course, a justification of the use of immunogenic epitopes, and since we hate to reinvent the wheel, what can be used in existing regulation can be used, should be used, and must be used, like, for example, all the compliance with material from biological origin, etc., etc. A very important part is, of course, the stability in time, the stability of constructs, and the stability on various temperatures. And last but not least, I want to insist on that. What we see with the new platform technologies like the MPSP in particular, where we have now a lot of data coming out, it's a vaccine by design. It's almost a mathematical description of a vaccine. where you can totally comply with the 3Rs principle, and why not go for a total in vitro quality assurance, quality and quality control for batch release and consistency approach for the production, which can be increased a lot compared to classical production methods. It's clear that this is the general framework and that specific requirements are to be determined per product and, if needed, per platform in what we call the annexes of the regulation as it is today. So let's go back a little bit to COVID now. On March 18, the ICMRA, an association of major regulatory authorities in the world, and most of them are represented today. So I cite literally what was written on March 18, 2020, opportunities to leverage knowledge accumulated with platform technology should be considered to accelerate the development of a SARS-CoV-2 vaccine manufactured using the same platform. In 2014, we could not imagine to have foreseen so good. If a platform technology utilized to manufacture a licensed vaccine or other investigational vaccines is well characterized, it is possible to use toxicology data, etc., and clinical data accrued with other products using the same platform to support first-in-human clinical trials for a SARS-CoV-2 vaccine candidate. And I think this is a fantastic argument we can also use towards those who doubt about the speed with which, on a justified way, the COVID vaccines have been developed. So the platform master file and COVID existing platforms, since they are already validated, no need to redo the complete file. The use of platform data used and offer a huge opportunity to concentrate on a new insert instead of redoing the whole process. And we have the feeling it could be extremely useful, this principle, for the strain variants where many vaccine developers and producers are already working on. I thank you very much for your attention, and I look forward for your questions during the discussion. Thank you.
Thank you, Joris. Just for the audience, all your questions will be selected during the roundtable discussion. So we are going to the next presentation, which will be given by Adam Hacker from CEPI, and he is Global Regulatory Affairs Manager at CEPI. Adam, the floor is yours.
Great, thank you very much. And can you see my screen?
Yes.
Yes.
Perfect, thank you. Thanks very much for inviting me. And I think it was very nice to hear ZAPI's progress that they have made in enshrining some of the concepts of platform technologies into the legislation, which is extremely important. And I'm just going to expand on that. So it's been quite an extraordinary year. I think the public health emergency was only declared back in January. And over the course of the subsequent year, we've had multiple approvals. And importantly, a lot of the agencies already have in place mechanisms to accelerate approval under emergency legislation. So we were able to see on the 2nd of December of last year that the MHRA under Regulation 174 was able to provide the first approval. On the 11th of December, the FDA under 564 approved 316 days. And then on the 21st of December, the European approval followed. Now that is a full-blown conditional marketing authorization, which is a traditional approval under the existing legislation. So it's not an emergency approval, so it utilizes the existing mechanisms to still achieve what is quite an extraordinary target, achieving an approval in 326 days. So a combination of extraordinary efforts taken by developers, the mechanisms that the regulators have in place to provide some level of flexibility to accelerate the reviews has led to this quite extraordinary achievement of approval in around 300 days. If we look at what has happened in the pandemic over the course of that 300 days, it's not a pleasant view. We've seen the typical exponential growth, both in cases and in mortality, 70 million cases and 1.6 million deaths. And the shape of the curve is typical. It's going into that exponential phase, and we're certainly in the exponential phase whilst many countries are locking down and taking extraordinary measures impacting their economy and the health of their citizens, mental health of their citizens, in significant ways to try to control the epidemic. But what would it happen if we were at a much shorter time frame to achieve a vaccine approval? If we could do that in something much shorter, such as 100 days, the numbers of people infected and the number of deaths would have been dramatically different. But importantly, the growth curve in the infection perhaps is in a different phase. It's not in the exponential phase. And by the time vaccines are introduced, which obviously has a lag after the approval of 3 to 6 months before we start seeing the impact on the population, it means that we're really controlling the epidemic and trying to keep it outside of the exponential phase. So the key question is, how can we ensure that we can accelerate vaccine development beyond the typical 10 years it has taken in the past? And of course, the regulations and guidelines are not really adapted to this environment. The approvals are granted for vaccines, they're not granted for the platforms. But we have seen over the last years that companies have looked at the data that they've generated. They're using common manufacturing processes, which they have experience for, and they're using particular types of technology, which they've built experiences over the years. The regulators themselves have adapted their regulations and ensured that there is flexibility with them, and that's often driven by the emergency situations. We saw that to a certain extent in the Ebola outbreak in West Africa in 2014, where it was possible to rapidly approve clinical trial applications, look at ways of streamlining the data for those approvals so that products and clinical trials could be initiated much faster than would be anticipated. We just heard about the ICMA meeting in March of last year, where there was a general discussion about the ability to use platform data and technology to accelerate vaccine development. And importantly, the FDA issued a guidance in June of last year, which started to really hone in on the principles of what developers needed to provide and where they could rely on vaccine experience. So what are these areas? Well, I think platform data can be used in certain areas. Companies have experience with process validation, process controls, quality, critical quality attributes, and in many instances, those don't have to be modified significantly, and the amount of data to support a new vaccine using those same technologies can be streamlined. Is the full-blown repeat dose tox study needed, at least prior to initiation of clinical studies? That's an important data, which often is required and yet can slow down development quite significantly if that data is needed. Can you utilize it in order to get into the clinic sooner? Is there experience from clinical data which allows one to get into the first in human study faster, pick a dose, at least minimize the number of doses which are being looked at in those initial clinical studies, and in particular, ensure that you can accelerate into high-risk populations such as the elderly. Of course, that doesn't mean that vaccine-specific data isn't going to be required. It certainly is. Clearly, elements of potency and identity, possibly some level of stability, although preferably being able to cross-refer to data on longer-term stability to give a better shelf life and avoid some of the complexities and difficulties of pushing vaccine product into the market, which we're starting to experience now, whereby there's very short shelf life on product, and that actually is impacting supply. Some level of non-clinical data is going to be required, certainly the proof of concept to demonstrate that the product is working, possibly in parallel to getting into the clinic. And a certain level of safety and efficacy is always going to be required. We obviously cannot compromise those two important hurdles. And the key question which we have to debate in as part of this workshop is just how much data is going to be required. So we've sort of gone from where we were a number of years ago, where everybody was 10 years was the normality. I think over the last few years, we've been accepting that things can move a lot faster. But if we had been sitting where we are today, a year or two ago, for us to say that we could get to a vaccine within 300 days, I think everybody would think this is quite revolutionary and quite extraordinary, and yet it has been achieved. And I think we have to certainly build on that. And the key question is, how can we build on that? Now, the emergence of new strains of coronavirus is, of course, of great and deep concern, but it's also acting as greater thinking process to really unravel where we can maximize the experience of the existing, the most recent approvals, to be able for them to pivot into to create a vaccine targeted at new variants. And here lies an opportunity to really do that extremely rapidly, and of course a necessity to be able to do that. There is certainly a rationale here in this specific case of minimizing the amount of data that is going to be required. Products that have already received licensure, when they pivot to a new vaccine against the variant, they will be using the same manufacturing process and controls. Inevitably, they will hopefully be using the same manufacturing facilities. A lot of preclinical data is just not going to be required, and I think the regulators are indicating that is the case. Vaccine efficacy studies won't be necessary. Those 40,000 subject studies, which took significant time, albeit rapidly, to accrue, will be faster because there will be an ability to bridge across the previous approvals from an immunological basis. And whereas previously the regulations have really required some more than 3,000 subjects for a new vaccine, in this situation where we're pivoting to the new variants, less than 3,000 subjects is going to be acceptable. And it was very good to see that the European Commission issued a press release just yesterday about the HERA incubator, where they're really looking at the regulations as how vaccines can be adapted, or new vaccines can be put into the market, whereby we can speed up the approval process. So we all have the tools in place to be able to do that, and that will act as an important test case in how quickly we can move. But it doesn't detract from the fact that there will be another epidemic, pandemic around the corner. COVID-19 has shown just how devastating such a pandemic can be to the world. And therefore, I think we have to reflect on how we can utilize platform data to move things much faster, even faster than we've managed to achieve. And that means really being able to accelerate vaccine development, really use the platform experience that we have gained during this pandemic. And CEPI's ambition is very clearly to enable this ecosystem to work with regulators and product developers in order to accelerate work to be able to move from the establishment of that new pathogen sequence to get it into the clinic as quickly as possible, and really to get an approval against that future disease X in an unprecedented time. So whereas we were looking back a couple of years ago, we might have said 300 days was quite extraordinary. I think with the flip to new strains, where we might be able to achieve that in a much dramatically reduced time of perhaps 100 days, where will we be in the future? And will we be able to establish a regulatory process, a platform level of experience which will give us confidence to be able to move to getting pivotal, important clinical data in 100 days. It is a significant challenge, and it may take us a while before we get there, but it's a very important goal to achieve so that we can manage effectively through that pandemic curve and avoid getting on to the exponential phase and the damage that has brought the world. And with that, I will end. Thank you very much.
Thank you, Adam. And all slides will be available after the meeting on our website, and we will also stream this whole webinar so that it can be reviewed later onwards. The next speaker is Martin Friede. He is the coordinator of the Initiative for Vaccine Research, Immunization and Vaccines at the World Health Organization in Geneva. Thank you, Martin, for being with us, and you have the floor.
Thank you. Let me just make sure. Can you see my screen?
Yes, we can.
Okay. So thanks very much to Peter and to Joris for inviting me to this meeting. I want to emphasize, I lead the vaccine research unit at WHO. So we focus on the vaccine research, not on the regulation. And what I'm going to try and talk about here, I fully agree with what Joris just said. I fully agree with what Seppi has just presented. Absolutely agree. I'd like to present a bit of a scientific perspective, especially seen from the point of view of influenza, where we have been using essentially a platform technology for many years. And from this, some thoughts just about some precautions as we go forwards. I do not want to talk about COVID. WHO is right now in the process of developing guidelines for how we incorporate the updated sequences into the platform technologies. I do not want to mention any aspects of that because that could interfere with the discussions. So I'm going to focus on flu and some general thoughts. So let's get into this. Now, the pros and cons of vaccine platforms. We've heard about the pros. We've got production processes are validated. The CMC is very similar. We don't have to redo the entire package. Essentially, this means we can speed up the process and submission of CMC. And in many cases, we can even avoid efficacy trials. We can go to immunogenicity trials. We don't need to go for big safety trials. But I'd like to bring up a few issues that we've run into in the past. How much does sequence change impact the process? To what extent is the process standard? How do different inserts impact the stability, the safety, and the immunogenicity of the vaccine using the platform? And very importantly, something not widely discussed is how does prior exposure to the platform impact the safety and immunogenicity? And I think these three areas, which I'll go now into some detail, we need to take these into account as we consider using platforms for the future. There is no doubt the platforms bring a huge advantage in speed, but we need to be considering some of the science behind this. So just a few words about the clinical experience we already have with platforms. Influenza is, of course, the best example, I think. We've been using the same influenza process every single year for the last 70, 80 years. We choose in about February for the Northern Hemisphere what the upcoming viral strains are going to be, and 9 months later, the vaccine is distributed. There is no time to do safety and efficacy study. We're using the same process every year. We're simply changing the strain. And in most countries, a small immunogenicity study is adequate. And in some countries, that is not even required. Now, what we've seen is that this also has applications in response to the pandemic. So in 2004, when the H5N1 scare started, a number of platforms, and in Europe, these were mock dossiers, were presented. And so when the pandemic, the 2009 H1N1 pandemic broke out, these were put into place. So we were able to get very rapid approval. It was 9 months from the discovery of the virus to approval. It was actually only 5.5 months from the day we announced the public health emergency of international concern through to the moment the vaccine was approved. Very, very fast. And this included adjuvanted vaccines. So a clear example of a platform being able to adapt very quickly to a new strain, to a new outbreak. But let's just for a moment talk about the use of this platform for seasonal vaccines and the question of how well do we really understand influenza platforms, for which we've got, as I said, 70 or 80 years of experience. There are essentially 4 platforms: egg-derived split, tissue culture-derived split, recombinant hemagglutinin, and live attenuated. So a few words. Let's just look at this slide. This is the vaccine effectiveness to live attenuated influenza vaccine, or the effectiveness induced by the live attenuated influenza vaccine over a couple of years. Now, this vaccine has been used in the USA for the last 25 years, 30 years or so. And every single year, it was giving vaccine effectiveness in the region of 50 to 70% in the young children, 2 to 8, and slightly lower in the slightly older age group of 9 to 18 years old. Everything was going perfectly well. Each year, we were simply inserting the new strain into this vaccine, and we were getting good effectiveness until suddenly in the 2013-14 year, in the young children, the efficacy dropped from 60, 70% to a negative 31%. What was this about? And interesting, at the same time, in the older children, we didn't see much of a change. Now, it would take me an hour of presentation to walk you through the data behind this, but I'll come down to the crux of it, which is in this slide here. What had happened is that this technology was developed and the platform was developed before the H1N1 pandemic in 2009. And all of the strains that came before 2009 had an affinity for an alpha-2/3 sialic acid binding. This is getting into the details. In other words, these viruses bound very well in the nasal mucosa. But the introduction of the porcine H1N1 sequence in 2009 resulted in a change in the hemagglutinin binding. It bound preferentially for the H1N1 strain to an alpha-2/6. Sorry, it says 2/5, that should be 2/6. And this means that this viral strain did not bind very well to the nasal mucosa, but bound lower down in the respiratory tract, partially explaining its pathology. This was not picked up because no quality control tools had ever been needed to look for this. So we had a platform that had been working for 30 years nearly, and suddenly it stopped working. Now, we went through a lot of studies to try and find out why it stopped working, and we realized that the insertion of a new sequence into the platform could have a major impact. Now, later on, I'm going to be talking about tropism. Essentially, what happened here was that this glycoprotein from the pandemic H1N1 virus had a tropism for the trachea and not a tropism for the nasal epithelia. Since then, we've been able to adapt the H1N1 strain of post-pandemic, and now we've been able to get H1N1 strains that once again have a tropism for the nasal epithelia, and so we've solved that problem. But just as a warning, that this is an example of when the platform does not readily accept a new insert. Now, another topic which is causing us some concern, which is something which comes by various names. Usually it's called the original antigenic sin. This was a term that was invented in the 1940s. And what this graph is showing you here, I don't know if you can see my mouse, but if we look at the third line, these are people, the third line down, where we've got the number 65. These are people that received the vaccine this year, but did not receive the vaccine over the years before. In other words, they're relatively naive and they received the vaccine. And we see 65% efficacy for the H3N2 strain, 75% for the influenza B strain. But if we then go to the top line, which is people who received the vaccine this year and people that received the vaccine every year, such as healthcare workers, we see that the efficacy has more than halved. And like this is true both for the H3N2 and for the influenza B. Now, what's going on here? We don't fully understand it, this repeat immunization hypo-responsiveness. But we believe that what is happening here is that as we keep using the same platform, we are inducing immunity against the common denominant. In this case, the common denominant is a part of the hemagglutinin stem, which does not provide any neutralizing or any beneficial immunity. And so when we keep giving the vaccine, we, instead of getting the immunity to the relevant part, if we talk in COVID, this would be the spike or the tip of the spike. Here we're getting immunity to another part of the molecule, which is not bringing any health benefit, possibly even a deleterious effect. And so this original antigenic sin, or immunity to the dominant, to the pieces of the antigen that are being presented every time you use the platform, this needs to be taken into mind if we consider using the platform frequently. Now, just a few words about other things that we've seen over the years with influencer platforms. If there are people on this call that have been working with any of the big manufacturers, they might smile or grimace as I mentioned this. So those that are working on recombinant hemagglutinin, it took 20 years from phase 1 to approval. One of the reasons was, as we modified the hemagglutinin sequence every single year, the downstream processing had to be modified, and the range of things happening here. Isoelectric point change, so the ion exchange process had to change. We were seeing aggregation, again, isoelectric point. Then the Australian experience with the inactivation and splitting of the influenza, it had been working perfectly for many years. And then one year, a strain came along where, well, we're not quite sure why, at least certainly I'm not quite sure why, but the splitting was no longer complete. And so the vaccine contained a small amount of unsplit, this whole virus, completely dead, inactivated. But in young children, the residual RNA inside these whole viruses was now able to trigger a very severe adverse event in these young children. Fortunately, not frequent, relatively rare, but a severe adverse event. And so this vaccine suddenly had to have its age restriction changed from 6 months and above to 9 years and above. And then those of us that work in the flu field will remember a few years ago, a major European manufacturer had flocculates in the vaccine. And this happens occasionally from year to year. We see that we've introduced a new strain, and with the new strain in the platform, it's either aggregating or precipitating or flocculating. And while not a serious health issue, we don't really know how this impacts safety or immunogenicity, and these batches were withdrawn from the market, resulting in insufficient supply, shortage of vaccines, and the problems associated with that. So just a few issues here. Clinical concerns. Maybe I'm being a bit overboard saying concerns, but these are issues that I think we need to be conscious of when we talk about platforms. The first is repeated use and the immunity to the platform components. So one of the platforms we're discussing during COVID is, of course, the viral vectors. Now, the immunity to the vector, we suspect, may decrease the response to the insert. We've known this for many years with the adenoviruses, that in populations that have been exposed to wild-type adenovirus, they have a lower immune response to the viral vector carrying a neoantigen. So the question is, will we see this as populations get repeatedly immunized if we were using one of these vectors, be it AT5, AT26, MVA? Will the immunity induced by 1 year of use of the vector mean that the next year that platform can't be used? I think we don't know, but it remains to be seen. We need to look for the evidence. Then we've got this topic I discussed, the common denominants, original antigenic sin, boosting of a common non-neutralizing antibody. Well, we've seen with influenza that this can cause decreased vaccine effectiveness. But one of the things we've been worried about with other diseases, dengue was the one in particular, was the risk of an antibody-dependent disease enhancement. So again, we're not saying that it's going to happen, but it is something that we need to look for. And then something which has only recently been raised is, what is the risk of, if we're using the same formulation repeatedly, that we can actually be inducing antibodies to some components of these vaccines that we've never seen previously, because the vaccines were only administered in the first year of life and not later. So this is hypothetical, and issues brought up here, of course, polyethylene glycol, dextran. And then there's the issue of modifying the cellular tropism, which is probably only important if we're talking about replicating vectors. So if we insert a viral glycoprotein that has a tropism, for example, towards neuronal cells, will this take this replicating virus from being totally safe and infecting predominantly epithelial cells? Will it now start infecting neuronal cells? Now, this is not just a theoretical concern. We have seen with VSV, vesicular stomata virus, that as we insert different glycoproteins from different viruses, the tropism changes. And in the Ebola vaccine, we saw that the VSV actually acquired some tropism for synovial membranes, and we were able to isolate the viral vaccine, the vaccinal virus from within synovial fluids. This means that safety cannot be entirely predicted. And if we're going to completely change the glycoprotein using a new virus, we need studies. So just some thoughts in view of conclusion is that these technologies can, of course, greatly accelerate development. The genetic inserts into vectors or nucleic acids, these are game changers for how we're going to respond to outbreaks. But not all vaccine platforms are suitable for all situations. We need to be monitoring how these things are happening and gathering an evidence base. So for the moment, we have to go as quickly as we can, but ensure that we're gathering the data. And I'd like to mention that innovative QC tools, quality control tools, may permit identification. So I'd just like to finish on one last thought, that for preparedness, the world needs the platforms to be ready. So we've seen from our thoughts around pandemic influenza that we can't build a facility that's just waiting for an outbreak. That is not sustainable. So what we need are platforms that are being used on a routine basis. So we need to find a match between our outbreak, for the platform we're going to use an outbreak. How do we use that platform on a daily, weekly, monthly, sustainable basis? And here we're looking for sustainability matches. And our first thoughts are mRNA. Well, maybe you could use this for seasonal influenza, but are we going to see repeat administration issues? Papillomavirus, polio, tuberculosis, a lot of people working on it. HIV, will this finally give us an HIV vaccine? Viral vectors might be more problematic, depends on how this immunity to the vector plays out. For the recombinant proteins, clearly this is going to give us a platform to test, evaluate, and produce adjuvants at a large scale. And for the whole inactivated viruses, which are showing some promise, one of the concerns is, of course, going to be, depending on the virus, does this require BSL-2, 2+, 3, or 4 facilities? And are you going to be able to sustain these facilities over time? So final thought is, will the platform work for the next outbreak? And my thought here is that I really think that we need better understanding of the immune responses to the platforms that we're working on right now. And I really hope that we can also find a mucosal platform, because for many pathogens, mucosal immunity might be appropriate, such as for live attenuated vaccines. And it can also potentially be better at reducing shedding than our systemic vaccines. I stop there.
Thank you, Martin. As announced, we will take the questions in the second part of the afternoon. Also already said, the next speaker had to apologize. He was not able to take part in this discussion this afternoon. So we go directly to Stefan Müller from CureVac. And he will talk a bit about the experiences CureVac had during the development of the COVID-19. Stefan, thank you. The floor is yours.
So thank you, Peter, and thanks for inviting me. So I'm trying to open up the presentation. Hope you can see this one.
Yes, we can.
Yeah, we can. We see it clear.
And now you see, I guess, the wrong.
Yeah.
You need
Correct.
to go to the other one.
Okay, now it's good. Oh, okay,
Yeah.
good. Thanks. So it's not really a presentation. It's really about a paar points, really for discussion that I have on our experience, also on the process, on the development process, but then also the process on how we then got to the next steps in the clinical development, and then at the end, hopefully also to the approval. So I need to put this in here, of course, as the forward-looking statements, and I would not expect that you read this. Really only 2 slides for me. So, and we heard a lot about the platform approach, and I also really liked Joris' presentation on this platform master file, and also how to actually include new technologies into this master platform file. And if you look at the mRNA technology that was now mentioned a few times, for us and also for the other manufacturers and developers of mRNA-based technologies, is of course 3 pillars that we have. So one is, which is also true, of course, for any vaccine developer, this is the optimized antigen design. Then, of course, what comes as the platform or the backbone is the optimized mRNA sequence, which is different to actually the antigen design. And then there's, of course, the delivery system. And we also heard about that there might, of course, also be some risk associated if you always use the same delivery system. But if we then go into the fact of we have variants, we want to be quick, and from Adam, we heard this to go down to 100 days. This would, of course, be excellent if we could achieve a 100-day, let's say, time frame to have then a variant vaccine dosed to people. Then what could we do? So for the optimized antigen design, this would be, for example, we have a good design. This is in the case of SARS-CoV-2, the spike protein. So this is used throughout and has been shown that it's really successful with high efficacy. So using this full-length spike protein, and there, of course, we could then adapt the sequence to the variants. Then, of course, the question is which variant to use, or is it a combination? Do we also include, let's say, predicted mutation that may come up over time? But this is a change adaptation of the coding sequence, and this then would call out for the specific variant. And that would then be a fact here, and this is a schematic of the entire mRNA sequence, and one part of this is the so-called coding sequence, which then gives rise to the antigen. So we would change here little bits of this coding sequence. The rest, this is the 5' cap, untranslated region, and also, of course, in the 3' end, also the untranslated region and the poly A tail, this will stay constant. So if you look at this one, you have, in principle, you have 100% of identical constituents. You have a change in the sequence of way less than 1% of the nucleosides. So that's actually also fairly constant, with the exception of the specific mutations that we have. Then if you look at the delivery system, then of course you also would want to keep the delivery system identical. So we have more or less everything is the same in this platform, and that's also the reason you can call it platform, but you have these single mutations or deletions within the sequence that then would give rise to the modification of the antigen. But as a whole active substance, as a whole drug product, it would be extremely comparable across, let's say, the wild type versus the sequence. So the question is there any preclinical testing required? Is like with the seasonal flu approach, is there really the need for only one tailored comparative phase 1 trial, for example, where you compare to the Wuhan sequence, you measure immunogenicity, you measure it against the various strains that are currently circulating, so to prove that you have activity, and then you would allow with this data set then approval. The question that I would then have is, what is if, for example, this is new technologies? So every year, every company would have maybe an improved backbone that has a slightly better features. So the question is, if we have an improved backbone of the mRNA, what does this mean to the approval process? Would we then need to go the whole 9 yards with all the studies, or could we also bridge from the existing backbone to the new backbone by limited immunogenicity and safety data? And with this, I just go to the next slide. This is now for the panel discussion, also with the experience that we have from our development of our CVnCoV, so the SARS-CoV-2 mRNA vaccine candidate. So, of course, for everyone, it was really important to have early and frequent interactions that facilitates then also the acceptance of the data packages, and also facilitates to modify, let's say, the data packages that are needed. Of course, the more interaction you have, the more feedback you also will get. And some things we, of course, experienced is that, of course, if you go to different regulatory authorities, like in Europe, where you have different authorities, when you do studies in country A, B, C, then you may also get, let's say, a little bit of diversity of feedback. And that, of course, doesn't help speeding up the process. So this is, of course, different to, for example, if you go to the US, where you have then the FDA's one single agency. What also worked very nicely is the extrapolation of data from similar vaccines. This goes back to the platform approach that could be acceptable based, of course, on scientific justification. Also, for all the agencies we worked with, there was really outstanding flexibility and also responsiveness to address the situation, even being quite patient also with us as a younger biotech that is maybe coming quite frequently with questions to the agencies. Of course, the review timelines have also been greatly accelerated, and that was also one of the discussion we're having, right, from 10 years to 300 days to 100 days. That, of course, means that the review timelines have been greatly accelerated. But also here, if you have different agencies, for example, to start a clinical trial, you may also have varying review times of the authorities for example, time-sensitive input. Nevertheless, there's of course also within Europe a cross-agency interaction which addresses actually these, let's say, points that maybe can be improved with consistency of the responses, also the timing of the responses, and all things considered. I think these two platform approaches really are feasible, and we can then, I mean, we hope really that we come from this plug and play to really adapt the sequence and then also vaccinate the people. And with this, I would then also end the presentation, and this was really meant then to open up also the discussion that we had.
Thank you very much, Stefan. We'll go directly to our next speaker, Ruben Rizzi from BioNTech. So also a company who was able to come up with a COVID-19 vaccine quite rapidly. So thank you for sharing your experience with us, and you have the floor, Ruben.
Sure, I'm going to share my screen right away. Are you seeing the right screen?
Yes, it's perfect.
That's perfect. All good. Then thank you for inviting me, first of all. Just before starting, as usual, we have to go through some disclaimers on the product. And of course, these are just my views and do not reflect the position of BioNTech or any other entity. What I would like to discuss with you today, first of all, a little bit of introduction where we are in terms of definition of platform technology for vaccines, and a few words on our project, BNT162, or Lightspeed, as informally called, program for a COVID-19 vaccine. I would bring just a couple of examples, very high-level examples that I believe can be helpful also for guiding our discussion afterwards. And I would like to raise a few topics that maybe can be of interest also to understand how platform approaches can be implemented more frequently and for different vaccines development, not only in case of pandemics. This is a very good starting point, I believe. We have seen this wording already earlier during the webinar. It's from the March 2020 ICMRA meeting. This is the outcome, one of the outcomes of that meeting, really pointing at a direction where in case a platform technology is well characterized, then we can make reference to the existing tox data or clinical data in order to facilitate and support the first in human studies for vaccines for SARS-CoV-2. This was a very clear guidance suggesting us, the developers, to look into potential platform technology approaches to accelerate the first in human trials. I think there are also question marks here that are highlighted by this kind of sentence. For example, what is exactly the definition of this platform technology? This is not clear. I think we all have in our minds our own ideas, but still we are lacking a global, comprehensive, overarching definition from a regulatory standpoint that tells us what is considered a vaccine technology platform. And again, this is something that can be explored further, but also the regulatory framework in general needs to be adapted to some extent. As developers ourselves, we still think of a new vaccine as a product, a self-standing product entity, and not a platform that we need to develop. And when we obtain a license to market, we obtain a license for our product. There are lessons that can be learned, and I think we have already heard some very interesting discussions earlier today from the world of vaccines for flu, pandemic and seasonal flu. They are already introducing some platform or platform related principles that I think should be leveraged more, even in different settings. Another very important thing that I would like to stress here: RNA vaccines are a quite peculiar technology, and compared to other or more traditional, I would say, technologies for vaccines manufacturing, that can be really a good benchmark for exploring more in detail what can be the potential, the full potential of a platform technology approach. This is because it's quite obvious, but with mRNA vaccines, you have a very high predictability in terms of the manufacturing process, and there's a very close connection between what you can plan beforehand and what, especially in terms of drug substance, I would say, and what is going to be the actual mechanism of action in vivo. So you do have quite a scalable and reproducible process, and you can have quite a good control on this process. This is why, and I will show you also in the next few slides, how this can be particularly helpful when it comes to making reference to potential platform approaches for developing these vaccines. A few words regarding our program. I'm not gonna tell you a lot, but this has been called since the very beginning, very informally, again, a light speed project for obvious reasons, I believe. We all agreed, and I think this is a very important premise here, internally with all our partner and stakeholders, the situation that we found ourselves in the first half of 2020 really prompted exceptional development strategies. And I think this is something I really want to stress, because what we're going to say today, and probably the discussions that we can have around what was done for this development, is not something that can be extrapolated immediately and applied tomorrow as common regulatory practice. I think what we can do is to see in these extremely accelerated developments under exceptional circumstances what could be done, and we can take some lessons that we can elaborate further to become more common practice in the future. But we should not expect that all the strategies that were implemented to allow a development under such incredible timelines can be used already tomorrow outside of emergency exceptional situations. A few points that I will elaborate in the next few slides, how BioNTech's prior experience was very important for us to being able to really accelerate, especially at the early clinical phases, this development, and the fact that we started our first in human trial with 4 different vaccine candidates. This adds a little bit of complexity to the early clinical phases, but this is also one of the reasons why we could be successful. And this is one of the key points where really using a platform inspired approach helped a lot to accelerate the first in human trials, in particular in this case. I'm not gonna guide you through the whole timeline of the project. This is very high level. I don't want to tell you about each single milestone, but just why we're saying light speed. The phase 1/2 trial, you can see here the milestone. The first in human trial started here in Germany with the first subject dose before the end of April 2020. And we had the first approvals at the beginning of December. We had emergency use in UK, emergency supply under Regulation 174 on the 1st of December. We had emergency use authorization in the United States a few days later. And again, a few days later, we had the first proper marketing authorizations in the European Union and in Switzerland. This means that in only 8 months or less, we could go from the first subject dosed to the first approvals. And even better, if you look at where we started thinking about this project internally, it was not the end of January yet. So in general, in less than 1 year, we could squeeze a full development until marketing authorizations could be granted. I just want to tell you as a bottom line message from my side, nothing magical happened in these 12 months. So there's no mystery or anything particularly fancy. But of course, a lot of hard work from all the people involved, including also those people working in the regulatory agencies. for reviewing and accelerating all the procedures as much as possible. But also a lot of small ideas or little principles that were put in place to try and accelerate every single bit. And the next couple of examples that I will give you today are really simple ideas that were put in place. It's nothing special. It's just how some principles that can be inspired by the platform technology approach could be helpful in our case in particular. The first one is how the previous experience from BioNTech could be helpful in accelerating, as I said, in particular, the early clinical phases we're talking about right now. When I joined the company, it was December 19. There was no Lightspeed project in place. And I was working at these projects right here that you see in the upper left corner, mRNA cancer vaccines. I was working as regulatory strategist for the FixVac platform, for the iNest platform. This is literally the BioNTech's background that we had in terms of pipeline. Why this is so important for us? Because these are the internal technologies that were already under clinical development for oncology programs. So you see on the left, different RNA formats, uRNA, modRNA, saRNA, which is the self-amplifying RNA, and tRNA, and then different formulations based on lipoplexes, lipid nanoparticles, or polyplexes. Long story short, RNA cannot be delivered without a proper formulation that can protect the RNA from degradation before it can do anything in the body. And at the same time, it needs to be facilitated in entering the cells. So these formulations are particularly critical. And BioNTech already had experience in using these formulations in clinical trials. Of course, in oncology, it's a different setting, but this experience was critical for us. These are exactly the same ingredients that were used then for the first clinical candidates of the COVID-19 vaccine program. So the same RNA formats and the same formulations that we have been already exploring in clinical trials in oncology. So this is BioNTech's prior experience. You see a number of different applications, you see a number of different RNA formats and different formulations, and how this could be translated. First of all, we had more than 10 clinical trials running already before starting the COVID-19 vaccine program, exploring different RNA lipid nanoparticles formulations in humans. And we already had clinical data available for different RNA formats. All these clinical trials running in oncology had already been approved, obviously, by the regulators. So we did already have experience with the FDA in the United States, with the Paul-Ehrlich Institute in Germany, and other European authorities for having reviewed these trials applications. And this is important because, of course, we already got feedback in the past few years, for example, on the key quality aspects and expectations from a regulatory standpoint in terms of being able to accelerate initiation of first-in-human trials with this type of product. In practical terms, what we did was literally to put together an investigator's brochure for this project. If you look at investigator brochures, I think the first one or two versions as well, or even the third one maybe, we had literally an extensive table summarizing all these clinical trials ongoing in oncology, all the exposure, the doses, the formulation used, the RNA formats, and providing a safety and tolerability summary for the investigators and for the regulators, obviously, of what were the findings in oncology subjects. This has been extremely helpful. It's not what you would call normally a platform approach, right? It doesn't correspond to the definition that we do have in our minds of what is a platform technology. But still, being able to leverage this prior experience, clinical experience and non-clinical experience, of very similar products being developed in a completely different setting has been critical for us. A second example, which is probably more fitting to the actual definition of a platform technology approach, is what we did for tox studies. Just to summarize the early clinical development that we had for our program, we had 4 different vaccine candidates entering the first-in-human clinical trial, encoding 2 different antigens of SARS-CoV-2 through 3 different RNA platforms. What you see down here is the 3 different platforms. These are the same that we were using in oncology programs, as I showed you before. And here you see all the possible combinations, right? On the right, you see the 2 antigens from SARS-CoV-2, the famous spike protein, and then the second antigen, only the receptor binding domain of the protein, and all the possible combinations, because we have encoded these antigens in all the possible RNA formats that we wanted to explore. So we did have a number of potential clinical candidates for initiating our first-in-human clinical trials, and we needed really to have a lot of flexibility. I think we have Ralph Wagner here from PI. He knows very well, when we started our clinical trials, we didn't have any idea what was going to be our lead candidate in the end. So we did need to have all the flexibility possible in order to support a first-in-human trial, exploring different candidates and having, of course, a package that could support this study. What we did in our case was to put together a platform tox study. So I'm sorry for I really stole these bricks from my kids' room, but I think this is a very simple principle to explain how it worked. We investigated tox with the 3 RNA platforms separately, obviously, and then we investigated the antigen effect on tox, comparing the data from 2 antigens, RBD, full-length spike protein, on the same RNA platform. We didn't see any difference. We could understand that there is no antigen-specific effect, and therefore we could extrapolate this data to cover all the potential combinations. This has been extremely important for us because we could support a lot of flexibility in covering early clinical trials for all the potential combinations of the 3 RNA platforms and the 2 different encoded antigens, with a very flexible tox program that could explore really what we needed to know in terms of being able to understand the profile of our candidates. A few conclusions. What is going to be the future and how the platform technology approaches can be used in our case for COVID-19 vaccines, not only ours, but in general, and then in a broader scope, hopefully in the future. The life cycle management of COVID-19 vaccines is challenging for obvious reasons. First of all, the pre-approval phase was extremely short, and therefore it's quite normal that the post-approval phase is quite cumbersome for everyone. So there's a lot of post-approval commitments. We are continuously generating data. And of course, there are all the uncertainties related to the biology of the disease. We still don't know exactly what is going to be the long-term effect, what is going to be the dynamic of the pandemic over the years. There's a lot of uncertainties. Even predicting anything in the next 12 months is extremely complicated. And we do have the topic of the emerging strains. The role that these strains will have in the pandemic is still unclear, and how the vaccines will be able to deal with these strains is something that we are discovering on a day-by-day basis. So it's literally a continued development in our case. A few ideas for our discussion afterwards. Strain changes, is there something that we can do leveraging the principle of platform technology in order to adapt the existing vaccines to emerging strains? Can we use the building blocks principles in order to update the antigen sequence without generating additional clinical data for every new variant strain vaccine that we generate? Which data will be required? We are having discussions right now with EMA, with the FDA, with the MHRA and other regulators on how to regulatorily implement the strain changes in case it's needed and when it's needed. We're looking at different models, of course, but it's going to be interesting to me to see if in the next few years, when we do have a better understanding of the disease and our products, of course, can we really leverage the platform technology principle to update these vaccines without going through generation of additional data? This is something we should consider. And in general, how we can approach follow-on vaccines. If we want to introduce an optimized version, if we want to explore additional potential formulations. For example, from a drug substance standpoint, in our case, we do already have clinical data with different mRNA platforms encoding the same antigen than our approved vaccine. And we do have data with the same RNA platform encoding different antigens. So how we can use these data to inform the future development of potential follow-on or improved vaccines, and how a platform approach can be used to generate multivalent vaccines. This is something that is being discussed for several different platforms. And I think RNA is a very good benchmark, as I said before, to explore these opportunities. Gaps that we still have from a regulatory standpoint. Of course, if we want to fully implement platform approaches in the world of vaccines outside of specific emergency situation or exceptional circumstances, we need to think about different definitions for the medicinal product itself, how new active substances are defined. We need to understand how the manufacturing changes to the backbone technology, to the backbone of the platform technology, can be implemented or can be handled and managed in the context of a platform approach. We need to consider what is going to be the impact on non-clinical and clinical implication, of course, but also from a data protection standpoint. This goes with the new active substance definition. To what extent we are still talking of the same product, to what extent we're talking of a new product, a separate one. This is something that needs to be implemented if we want a more broad and a comprehensive adoption of this principle. And pharmacovigilance, of course, do we need to think of a per platform reporting? This is something that currently is still to be defined in detail. Just a few conclusions, I'm done. I'm slightly over time, I'm afraid. Different platform approaches or platform-inspired approaches or principles have been really instrumental for us to accelerate our development, especially in the early clinical phases, I would say. And I believe the RNA technology in general is a particularly good benchmark. I've repeated this many times, but I think it's really true. It's intrinsic with the properties of this technology to explore the platform approaches. It's really it's a building blocks technology, and we can really try to explore together with the regulators to what extent we can push this flexibility. I'm pretty convinced that platform approaches will become more common in the future outside of emergency situations, but this will require new and clear definitions and, of course, new guidelines. I think that the examples that we have seen so far for these incredible developments for COVID-19 will be probably reflected in the next few years in a number of more well-established guidelines and updated definitions, and probably new ideas in general for all the potential development of new vaccines. Thank you, everyone, and in particular, thanks for everyone that supported this incredible program. I was just, you know, I've been lucky enough to be part of it, but it's been a privilege for me.
Thank you, Ruben. I'll let you go because we have a little bit more time due to the fact that
thank you.
J&J presentation has fallen away. We are 15 minutes in advance, and as some people will join after the break, I would like to take some questions first and then have a break at 3:30. Just one remark to you, Joris. Martin had to leave for half an hour. He will be back at 3:30. But maybe you can start with some questions to the other speakers. Thank you.
Hey. Thank you, Peter. So the first question was directed to me. So it's a question about the fungal platform. Can this be much faster, cheaper? And if this expression system can make a lot of therapeutics cheaply and quickly, that could be a super solution. So our ZAPI experience shows, and we were all positively surprised that this C1 system, without going into the details, shows that it is a high performance, can produce high quantities of apparently excellent antigens, very well confirmed, very well glycosylated, which has been confirmed in experimental animal models so far. So indeed, this is as an example, something we should look at with a very open mind and look at exploitation in the near future. Like I'm convinced there will be other platforms as well that will be coming. The second question is one for statisticians, which I am not at all. So it's a question about Bayesian statistics is inherently better suited for platform approaches than the traditional statistical approaches. So the question is, how can CMC statisticians who understand these methodologies get involved in these initiatives to show how adopting these methods and can support the goals of these initiatives? I don't know who can take this up from this panel. I don't know whether someone is a statistician and who can answer that. If none, I propose that, yes, Dean?
Yeah, Joris, like you, I am explicitly not a statistician. However, I have the good fortune of being pulled into these discussions through Tim Schofield and meetings he's organized through Iabs, and working also with both our CMC and clinical biostats colleagues. And I have to agree with the comment that Bayesian analysis seems to have much more capability to deal with multivariate pieces as well as rapidly moving through adaptive trials. Those are sort of the larger framework pieces. They do involve manufacturers coming in early and setting up those pieces. It's not something you throw in at phase 3, it's something you start at phase 1, but it is also adaptable to the CMC side as well. So it's a very good comment. It's something that's been brewing for a number of years, and people like you and I, Joris, are sort of being pulled into this loop. And it's one that's it's been quite fun for me to learn, at least sort of at a very high level, the potential power. And this is all made now much easier with modern computing software that can integrate these. But there is a real this has to be done by very competent people, well advised by those both in the clinical and CMC side as to what the realistic limits are of these things. And you have to be looking at this also very clearly in a risk-benefit model. You can't explore simply risks in clinical or any one parameter. You have to do this in a very balanced way. But there are many competent people to do that you and I can rely on. Over.
Thank you very much, Dean, for this explanation, which lightens at least a little bit up to someone like myself. Then I have two questions on the flu and particularly directed at Martin. So Peter, I propose to come back to then a question from Carmen Jungbeck and another one from Paul Mittling. So we keep that later when Martin is back. So, and then I have quite a lot of questions on messenger RNA. So the biggest challenge, Bruce Karpik, I think the biggest challenge will be that when we start development of an urgently needed vaccine for now, for a new indication, we will not know what the format or presentation will be efficacious, protein, vector, live, messenger RNA. Thus, we would need multiple platforms, both from a process of an analytical perspective, ready to go for the streamlined regulatory approval. This greatly increases the complexity of platform technology. So H1N1 flu was relatively straightforward at the time because it was already a universal egg-based platform. The main problem is, of course, the inefficiency of the egg platform. So the question is, in fact, how to deal with different platform approaches. And I would add to that the complementarity between them and the need, in fact, to have different solutions. Can we go to the Adam, Stefan, and Ruben to have an idea on this question? Nobody volunteering? So I would say indeed having different solutions, as we say, not putting all the eggs in the same basket, particularly fighting infectious diseases like those who have the privilege, like myself, who are in this field already since 40 years or more, is surely a good approach. Then I have another one from Koen Brusselmans. The CureVac presentation mentioned the need for consistent feedback advice from different regulatory authorities. Does this refer to different experiences between several EU authorities, or rather between worldwide authorities, for example, European Union versus US? Can
Stefan?
Yeah, I can take this one, of course. I mean, this is mainly as we did the studies in Europe and also in Latin America. This is mainly within Europe. This was the comment, and of course, it's also normal that different agencies have slightly. We receive, for example, for some rather questions on the CMC part, then on clinical on other parts, and then of course it's let's say less streamlined than if you have only one agency where you do your study. So if you do it in multiple countries within Europe, but also of course multiple regions, then of course it adds to complexity, and you have of course to address all the questions sufficiently. So this was the comment from my end. Maybe Ruben can also speak to that.
No, absolutely. I think this is absolutely correct, and it's the experience I think more or less of everyone to some extent. So I just wanted to go back to the previous question, Joris. Sorry, I was triple muted somehow. The one on the different platforms and the different technologies when a new pandemic comes. Of course, this is a very good comment in general, and you don't want to put all your efforts into a single platform, but this is a matter, I would say, more of public health more than for a single developer as we can be. So in our case, I think every company or developers or consortium has familiarity and expertise related to his own platform or the experience that it can have with his own pipeline or type of technology. I completely agree from a health interest and public health interest standpoint. It's important to have different candidates available and potentially reaching also clinical phase and late clinical phase, because you never know when you start a new pandemic, you really never know what is going to work and what is not.
Okay, thank you. So both to you, Stefan and Ruben again. Here, question from Dr. Gautam Sanyal. Should self-amplifying RNA be considered a slightly different or a sub-platform of the standard messenger RNA platform? So it's self-amplifying towards compared with standard.
We received exactly the same question from the Chinese authorities when we were initiating our clinical development. So it's a very good one. It really depends how we define our platform in first place, obviously. And so I don't think that for what we have seen so far, also in terms of early clinical data, there's room to say that it has a particularly different profile in terms of clinical profile compared to the other RNA platforms. But of course, this is a discussion. Let me put it that way. If we want to bring these candidates based on self-amplifying RNA forward into more advanced clinical development, and we reach the phase when we have to discuss with the regulators how this product would be marketed compared to the existing ones, this is going to be a very relevant question. As of now, we don't have a clear definition of what can be considered an RNA platform. So we do have these three RNA technologies that we consider as platform themselves, independent one from each other. I don't see a definition that can fit the three of them together. So I don't think as of now we can propose that we switch our antigen expression through the different platforms freely without generating any additional data. This is not the case yet. These are three different expression platforms, and what we can see is that the same platform has a consistent profile irrespective of the antigen, but not the other way around. So it's something that will need to be discussed in the future, but it really goes back to the fact that we don't have a definition yet on what is the platform that we're talking right now.
Stefan, do you have some to add to that?
Yeah, I mean, I can, of course, we do not embark on the self-amplifying, but of course, it's about what is really the platform, and certainly self-amplifying versus, let's say, a modified or unmodified mRNA, that's certainly some differences. The question to me that I also post on one of my slides is, what is if we simply change the backbone slightly, so it's more or less the same, right? Do you have, let's say, higher expression, or you have some changes in some of the characteristics, although you have the same type of mRNA? Is this then also the same platform, or what level of additional studies you would really need?
So that's a question to the regulators then. Okay. Peter, do we stop here? And then questions are arriving now. So
I think we should, Martin has arrived again. We take 5 minutes break here to get a cup of coffee or a cup of tea or something else. And we resume at 3:35. The floor to the panel members that didn't give a presentation, that they can present themselves and give their background.
Okay, thank you. Go on, Peter.
Okay, then I'll go like they are on my screen. Stefan, you are next to me on my screen, so can you just introduce yourself?
Yeah, thank you, Peter. Thanks for inviting me. I'm Stefan Tirstrup. I'm an ex-regulator. I'm a medic by background, but spent more than 10 years as a Danish member of CHMP and the Danish member of the Committee for Advanced Therapies at the European Agency, and also as head of licensing in the Danish Medicines Agency. I've been consulting for industry for the past 8 years.
Okay, thank you. Dean, you are the next.
Great, I think I'm off mute there, yeah. I'm Dean Smith, I'm with the Health Canada. I have experience both on the viral and bacterial vaccine sides, and I'm coming at this from a CMC perspective, although I have many good quality clinical colleagues to share and exchange with on this as well. Over.
Thank you. Rolf?
Hello, good afternoon to all. I hope you can hear me.
Yes, I can.
Yes.
Perfect. So, hello to all. My name is Ralf Wagner, and I'm at the Paul-Ehrlich Institute in Germany, who is a regulatory authority within the European network at EMA, and we have been very much involved in last year and also this year in giving scientific advice to COVID-19 developers in the approval of clinical trials, and also as rapporteur for licensure in the European system. Thank you.
Thank you. Marco?
Yes, hello, good afternoon. I'm Marco Cavalieri from DMA. I'm chairing the COVID-19 task force, and I've been following all the activities around vaccines and therapeutics for COVID-19.
Okay, thank you. So unfortunately, Marian Gruber was going to join us as well. Maybe she will join a little bit later, but she is, of course, also overwhelmed with a lot of work. So let's see whether she can come. And if she comes, then we'll give her the floor. Martin is here. So I think we can start, Joris, with the questions to Martin.
Yes. A first question to Martin. So flu platforms are more similar to classical vaccines, is the remark. So there are significant differences to the modern platforms currently used for COVID vaccines and those developed, for example, within ZAPI and other initiatives. Even if questions on safety, stability, etc., need to be answered, comparison between these systems is critical, and I should join. It's quite complex. And then I have another one on the flu vaccines. Certain platforms are permissible and usable across different species and used already for routine manufacturing for veterinary vaccines, for example, thereby securing continuous technology preparedness and manufacturing capacity. So the question here is, can we take more profit of experiences on both sides of the One Health border? Martin.
Yeah, so let me answer the first one. So I'm not quite in agreement that flu is similar to anything else, because flu is so far the only vaccine that we're giving repeatedly. Most vaccines, you get them once or twice in your life and we're done. So I think we have to look for flu to how platforms are going to respond repeatedly. But I will point out that the case that I brought up, which was the live attenuated influenza vaccine in 2013-14, this is no different to the adenovirus type of construct, because here we're using, in America, we use the Ann Arbor virus, which is a 1954, and in Russia, we're using the Leningrad virus. And we are simply we'd be doing a reconstruction every year of taking this 1954 virus and then putting into it a new hemagglutinin. So what I showed there of how suddenly one antigen doesn't work in the platform anymore, this can equally apply to adeno or MVA, probably not to the mRNAs, but certainly highlight that as an example of how a vector which is being used can be highly affected by the antigen that's put in. The fact that they're produced on eggs, well, we're also producing them recombinantly, we're producing them on tissue culture and less eggs now. But it's the only platform for which we've got years of experience of saying, how does this platform behave year after year after year as we keep changing the insert? Then the question on veterinary, I'd actually almost send this question back to Joris, because I've got a little bit of shortage of knowledge of this. What I would point out is that some of the vectors, again, the viral vectors, got a completely different safety profile. The VSV, relatively safe in humans. I don't think you would want to use this in equines. It would be fatal in equines. And I don't know about the bovines either. But again, I presume you're talking about a situation where the vector has already been shown to be safe and effective. But Joris, I will send that question back to you. It's much more your expertise than mine.
Thank you very much, Martin. So I will give a general answer, so not specific to one particular vector. So you know that, you probably know that in the veterinary sector, the pox recombinants have been used since more than 20 years already for almost 20 different diseases, whether in cats, dogs, equine. And the subunit principle is already known since a long time. For example, in the veterinary field, the production on cell-based flu vaccines in swine, in particular, is it's something classical. The question which is posed there, and I should say yes, couldn't we look more to each other? What experience exists, and from these experiences, try to learn in order to respond more quickly. So the MPSP that now has been developed in the ZAPI project, so there are many MPSPs in the world. So is another example. So can we use that? And I would add something else. When I see in the COVID discussion that until a couple of weeks ago, all specialists said, yes, we do not know whether the vaccine has an impact on the circulation of virus. No, it starts to be confirmed, particularly in Israel and elsewhere, whereas the non-human primate data were quite clear and quite indicative. It's never white or black or yes or no, but can we not use more of that? And I know that the regulators here have surely another position, but I just launched the question. Over. So then do I continue the questions, Peter?
Yeah, go ahead.
So a question here from Rongsu: can master platform databases be shared with vaccine developers? So it's more the producers of vaccines that are concerned here. As a developer, as part of ZAPI, I should say, well, the most interesting things are published, are in the public domain. So, but then what is more proprietary, that's a different question. I cannot answer that for myself. Is someone willing to answer that? If not, Dean?
Yeah, I think you answered it already, Joris. Is certainly from a regulatory perspective, it wouldn't be up to us to share that. That would be the manufacturer's willingness to share that. We are specifically in the business of keeping those secrets, so that's off our table. But if a manufacturer wishes to, that's a different story entirely.
So another question from Gautam concerning the quality and quality assurance and quality control analysis for batch release. So the two examples he gives is about release assays that have quite a long term and a long time lead. For example, cytopathic effect in cell cultures can be quite long as a potency dose determination for live attenuated vaccines, for example, and vectors and traditional sterility. So today there are alternatives which are faster, more precise. Why are these alternatives not yet used towards or not yet proposed to regulatory acceptance? So I think the regulators and the producers are concerned here. Who is the first?
Well, maybe I could start off with giving an answer. Ralf Wagner speaking. As the Paul Ehrlich Institute is also an institution within the OMCL network that is responsible for batch release testing. So, and therefore I can assure you that, of course, all the batches for the COVID-19 vaccines in Europe are tested by an OMCL that has been pre-assigned quite let's say half a year ago, having good discussions on the European level, also to investigate which OMCL can conduct specific assays, and also about the capacities to make sure that all the COVID-19 batches which shall be released for the market can be tested in a very short time frame. And also within this half year of discussion, the assays that have to be applied according to the EDQM guidelines, which are vaccine-specific guidelines stating which tests have to be performed for the release to the market. These assays have been established in respective laboratories during the last half year. And PI is, as an example, PI is at the moment the only OMCL testing the BioNTech vaccine and having released all the batches for the BioNTech vaccine on the market. Our Austrian colleagues are responsible for the Moderna vaccine, and the Netherlands, the Dutch colleagues take over the AstraZeneca. The tests that are performed for the release of such a vaccine are defined and established, implemented during the licensing procedure. So the manufacturer, of course, makes suggestions. They implement their testing program that will then be evaluated during the licensing assessment, and will be will also be checked whether all the assays are valid and whether the assays are relevant to confirm that the batches are compliant with the intended quality aspects. And at the moment, as was said, it is really it is true that for the viral vectored vaccines, for example, the titer of the vaccine is really one of the most important parameters, the potency parameter. But if a company comes up with a novel method and showing that this novel method is as good as for example, a titration, then of course it is not a problem, and it is really a rule that such a novel method can also be implemented. Thank you.
Thank you, Ralph. I see that Adam raised his hand.
Yeah, I mean, thank you. I think some of the big advances we've seen in assay release has come in the advanced therapies field, where particularly oncology, the sort of vein to delivery time becomes absolutely critical, and the necessity to really squeeze the timelines is absolute because the patients are dying. Therefore, things like sterility, rapid methods are working their way through. I mean, they've dropped from 10 to 12 days. You can do it within 5 days with the BacT-Alert system. And I think there are other technologies which are shrinking that down even further. I mean, you've got a couple of issues with vaccines which are important for consideration, and some relate to the fact that the products have short shelf life in the initial phases of rollout. And I think we're probably seeing that at the moment. We know the product is hitting the market with only a few months of shelf life. So additional batch release testing is going to cause a problem. It's going to mean that material is essentially could be going in the bin when it could be going into the population. And that's obviously of concern. Some agencies, as we've seen, have started to allow for paper release from a review once there's some level of experience delivered by the company on their batch release documentation, assays, etc. Then some agencies are allowing for that paper release. And I think the WHO has released a short paper a couple of months ago, or maybe it was even January, which outlined the criteria, which did give an authorization under the EUL to allow some national authorities to release on the basis of paper. And of course, that's important in some situations, not all situations. But I think the opportunity that these emergencies afford us is to really look at some of the fundamentals of what goes on with regard to batch release and try to identify ways that we can accelerate processes, accelerate the use and the trust in the company generated data to be able to do more paper release, to alleviate some of the bottlenecks in supply and short shelf life.
Thank you, Adam, for this clarification. Dean, yes?
I want to pick up a point that Ralph mentioned, which is actually very critical. So the question that came in was, why are those assays being used when faster ones could be used? And again, that's as Ralph mentioned, is up to sponsors to drive those innovations. Clearly, if data is presented that a better alternative and quicker test is suitable, then that should be put to any agency or collectively within Europe that would speed things along. So there's nothing in EDQM process that I'm familiar with, and Ralph already spoke to that would prevent that. But to Adam's point, is that some agencies have done things differently within the context of the current pandemic, and for different reasons, FDA and Health Canada have come to a similar position. And that is that based on looking at to Adam's point, the performance of manufacturers with their assays and looking at the consistency of manufacturing within in situ in their sites and facilities, we in Canada have what is still a formal lot release process, but we have risk managed it in this case, and are basically accepting the release of products based on the performance in-house within the manufacturers. We are simultaneously setting up assays to look at specific batches, but sort of heading down the road to formal licensure, because we are under an interim order process right now, which is more than emergency registration, but short of a full registration. But we are satisfied with the lead companies so far. This is a really important caveat. Because these lead companies are under stringent regulatory oversight, either within North America or Europe or other jurisdictions eventually with other sites coming online, that we feel confident based on the data they submitted to the review that we can do that in this situation for all the reasons Adam mentioned. So there are different approaches to this, but I know that many other jurisdictions do appreciate those OCOBRA certificates, and that helps them. So that's another element to consider as well. Over.
Okay, thank you. So in the same sense, we have here a remark from Carmen, so that accelerating batch release can be done by parallel testing. The procedures are already in place since many years. So, and I would join to that. So it's also the opportunity to mention again, if it's still needed, that the move from animal batch release testing to in vitro would also be a very helpful tool in order to speed up and also to increase the quality of the quality assurance, quality control. I have here an interesting question, I think, particularly to Marco. So the question is from Mats Wellen. Very interesting to hear about the concept of platform master files. Currently, the human legislation includes an option for vaccine master files, but these are antigen specific and not applicable to platform in general. Has there been any discussion at EMA, European Commission, on the introduction of such a master file concept? I believe this is at the human regulatory processes. Can I address that to you, Marco?
Yeah, I can start, and Ralph can complement as he's following also the Biotech Working Party. So, you know, there has been discussion since a while about what can be done here. And I think we have not really progressed on this, to be honest with you. But of course, it will be very important in the aftermath of this pandemic to really look into how far we can go with this option, and really understanding if there is any possibility to expand the current framework of using a master file. That's probably what I can say for the time being. I do believe that we need to have this conversation. We really need to understand how far we can go with this, and really thinking with an open mind how we can make things a bit more flexible and easier for everybody in light of the knowledge on platform technology.
Thank you very much, Marco. Any other? Yes, I see. Yes, Stefan.
Yeah, just to add to what Marco said, and I think it's, and to Ruben's point, that we first of all need to maybe agree what is a platform technology. That may not be agreeable across the line. It may be for different vaccines or different types of vaccines that we can agree what is encompassed in the platform, what covers the platform, and what's within the definition, what's outside. And then I think to your point, Marco, it's up to the sponsors, to the vaccines manufacturers to challenge the regulatory authorities in Europe, in US, and so forth, to initiate this discussion and to push for a scientific discussion and an agreement on what can we do now, and to the point that if we want to make vaccines in 100 days, or we even may need to make if we need to switch one of the current vaccines to a new antigen, we need to have this discussion going on now. And maybe what we can decide today is something different than what we can decide in years. Taking the WHO experience in with flu vaccines as an example, knowledge has been generated over the past 30 years. But at some stage, we need to take a decision and then, of course, follow things. But that's more common.
Okay, thank you. Dean, you had to add something. Yes, Stefan first.
Yeah, thank you. So just a quick follow-up on also the release testing. And if we talk about a platform technology, of course, what would, of course, be, I think, in the future an advantage if we have some of the key release tests that are valid for, I mean, just for this example, mRNA platform, they're valid for a BioNTech vaccine, as well as for a CureVac vaccine, as well for Moderna vaccine, that one lab is actually doing also the same, one of the key assays. that was also, I guess, increase, let's say, the confidence, and would also avoid a lot of back and forth. I mean, if you read also the IPA from Comiranti or also from Moderna, there's, of course, for some of the assays, there's certainly still some development work to be done, and the same holds true for us then, certainly. So this would maybe also facilitate some of the key release assay. Also eager to hear feedback of Ralf and Marco, and of course also of Ruben and others, whether this would be an approach that you could go.
No, in principle, I believe yes. We need to consider that in our experience, there's a number of aspects, including release, but not only that one, that could have been probably harmonized in a different situation, so with more time. There are a number of discussions that were a first time for everyone around the table. The regulators, sometimes in terms of timelines, even managing the contact with the OMSCL in a moment where we were really apparently far away from a market approval, honestly. It was difficult for us to figure out what was going to be the final aspect or the direction where we were taking. If we had known beforehand what were other competitors doing, other developers doing, I think in principle the room for harmonization is there, and try to converge for the OMCL testing, for example, to specific labs, or to align on what is going to be the panel beforehand in an harmonized fashion, that would be desirable in terms of optimizing resources and the time during the discussions during development, right? It's difficult as usual. I think everyone knows it, because in terms of development, we don't know what the others are doing. So, and the agencies, of course, and the people we're talking with cannot share too much for obvious reasons. And usually the alignment under such an intense time pressure is always complicated. We have seen this kind of discussion on many aspects, also in terms of, for example, the risk management plan. We were being reviewed in parallel in a number of countries with Moderna's vaccines. And we know there are some aspects that would have been ideal to align with them from a PRAC standpoint, from an agency standpoint, and even from our standpoint. But it was not possible, not because we were not willing to, or they were not willing to, or the agency was not willing to, but just because the timelines are what they are. At least they were in this case. But I think in terms of future development, Stefan, yes, this is something we can try to consider.
Thank you.
Ralf, I probably want to add something. I don't know.
Well, I can say a couple of remarks to that. Of course, in principle, there's some kind of harmonization from the EDQM guidelines saying these are the parameters which need to be tested by your OMCLs, which are normally appearance, which is quite easy, but which is potency, integrity, identity. So this is what has to be tested. But the assays the different companies use can be different. So, for example, I know that, for example, for the integrity testing, CureVac uses another technique as BioNTech. So it is difficult for us as an OMCL to foresee all these tests you use, and it's also difficult for the companies to align on methodology already at a very early or a very initial time point at their development. So therefore, I would think from a procedural point of view, it might be difficult to really align the assays that are conducted. And therefore, I think as OMCLs, we are always in the position to be able to implement the assays done by the companies as fast as possible. And this is something that requires that we contact companies as early as possible to get their SOP, to get more input in the methodology. From a very, very principal point of view, of course, it would be the best way if the OMCL would do an absolute independent test of the vaccine, maybe applying also other tests. But this is, well, what we have learned, it's simply not possible, all the more in such a situation as COVID, where we're trying to get our assays running as soon as possible to have an assurance by the OMCL testing that the batches which we receive for parallel testing, and I have to say this is something we of course have established here, that all the batches are in line with the specifications. So this is all I can say to that. Thank you.
Thank you very much, Ralph, and the others. So now I have two questions from Arun Aranashalam. So first question is about it's about the control. So the intracellular expression of protein through messenger RNA platforms emphasizes CMI. I believe he means cell-mediated immunity responses, but it will delay the analytical evaluation and release of vaccines. How vaccines should be expedited without losing the importance of CMI for efficacy? And I think the question means, how can it be done quicker if needed? And secondly, he poses also the question about the platform technology based on messenger RNA for multivalent vaccines, so such as flu. Are different mRNAs expected to compete for their protein expression and presentation, perhaps in the same set of cells or not? Need this competition to be considered for the cellular machinery? And looking back also for the, as Martin mentioned, the antigenic SYN phenomena. Who takes this up? Probably we start with the specialists of the messenger RNA.
I can give it a try to the two of them, then I will just hand over to the other panelists. Regarding the, if I understand correctly, the question regarding multivalent vaccines is about potential competition in terms of cellular machinery. Okay, I can tell. Of course, this is something that it needs to be explored further. First of all, we need to generate additional data if we want to go in that direction for any sort of vaccines based on RNA. What we do have is an experience with multivalent vaccines in oncology, expressing different tumor antigens, and in that case, we don't see. So far, at least, what we can say, we don't see this kind of issue in terms of the different RNAs competing for being translated. And actually, we need to bear in mind that the actual amount of RNA compared to the overall cellular machinery and the overall endogenous RNAs present is really negligible. So what we are providing is really a drop in an ocean of RNAs that are already present in the cells and then are being translated continuously. I don't think this is going to be an issue moving forward, but of course we don't have data right now to say that a multivalent mRNA-based vaccine can work efficiently in the same way. So it's something that is worth exploring. I don't see the problem right now, but I can rule it out. The first point was about CMI assessment, right? So I think we need to distinguish what is going to be the immunogenicity assessment during development and what is the testing for release. CMIs are an important evaluation, especially for our vaccines, of course. And this is something that we explore continuously in our clinical trials, and we keep generating CMI data that are, of course, particularly relevant. But this doesn't mean that we don't measure immunogenicity via antibody responses as normal, of course. But this is not necessarily something that has anything to do with the release or the timelines for the release of the vaccine or for the analytical testing of the vaccine. So we do have integrity and potency assays, but not necessarily encompassing cell-mediated immunogenicity assessments. So I don't think, if I understood the question correctly, I don't see these as a testing that stays in the critical path for the vaccine testing and release.
Stefan, and then I would like to pass over to the regulators on these issues, on Dean and Marco and Rolf. Stefan?
Yeah. I mean, as Ruben said, so for going to the last point with the release, of course, this would be not part of this package for release. But of course, if we have this, whatever immunobridging studies, then it should be part of this and is not, I would not consider this as time limiting because this could be done in parallel to any antibody testing. But certainly for batch release, this would be not part of the panel usually that you have. With the multivalency, that's also based on our experience. There shouldn't be really an issue in terms of RNA translation to protein if you have really different let's say antigens expressed. Of course, where there might be an issue if we talk about you have the real spike protein and you want to transcribe or translate different spike proteins, then of course there might be a biological issue because you might have some trimers that are really having funny structures that you don't want to have. But this has nothing to do with, let's say, the translation of the RNA to protein. But for multivalency, that also shouldn't be really an issue. At least also in our models, we have never seen any issue. Also, if you have cocktails, so there's no competition, so to speak, on that.
Yes. Who from the regulators takes up first? Marco?
Yeah, I can start by saying that, yeah, of course, for the multivalent, we need to test it, and it's good to hear, what colleagues are saying from their experience so far with the messenger RNA. So I would not be too much worried at this point in time, but of course, we need to see the data and to understand what is going on in this case, as indeed, the experience is still limited. For CMI, I can only concur that, it's not something that really has to do with specification, but even in terms of understanding from the clinical side, I have to say that we're still struggling to really have a good understanding of what is the role of CMI for these messenger RNA vaccines. And I was also struggling seeing that, like Moderna and BioNTech, they have different responses, which has to do with a number of aspects, like the timing of when this was measured, the type of assay that have been used. But I think it would be good if we have a better understanding of the CMI component for messenger RNA and how much they matter in terms of protection, because that is not really apparent at this stage, but it might well be that it plays a role. So it should be part of the entire exercise of the deciphering the mechanism of protection and what is really important for correlates of protection. So it's even if, of course, the level of antibody seems to be, more important, but nevertheless, at least for prevention of severe disease, it could have a really importance also CD4 and CD8, and whether some more work can be done on the clinical sample would be extremely helpful.
Okay, thank you, Marco. Ralph or Dean, do you
Ralph,
have?
why don't you go first and I'll follow.
Actually, I don't have too much to say to that. I just would like to stress the concept maybe of the OMCL testing, which is not meant to really show clinical efficacy, but the concept is that we have a product that is tested for specific quality specifications. But then during the licensing process and the clinical trials, it has been shown that the product that has been taken to the clinical trials with this same quality specification has the intended efficacy. And that this is why we test, but as we test as an OMCL, we only test quality-related parameters, which have an indication from the process before and evidence also for vaccine performance. But we would not be trying to test CMI as a release test. Thank you.
Thank you, Ralph.
Yeah, I'll echo that, and it's been said several different ways, both by the manufacturing side and the regulatory side. Is that CMI is critical, and it is, as Marco knows better than I, it goes well beyond B and T cells potentially. It's a sort of mysterious immunological phenomenon at this point that needs to be better understood. But those are our clinical questions. They have, as Rolf says, they're connected through their critical quality attributes on the quality side, but for release, mRNA integrity, as Ruben and Stefan have mentioned, is really critical for that to be the case. As long as adequate integrity and cap and poly-A are in place, these molecules will work as they're demonstrated in the clinic. So it's that link that Rolf mentioned is critical. We certainly wouldn't want to see what are very important characterization assays on the CMC side, whether they be integrity, cap, et cetera, or I would say a really critical assay that would have helped in a lot of cases are these in vitro translation assays, which allow us to link up all of those pieces, know that we have functional molecules. These are very good characterization tools in addition to cell expression, but they're characterization. They're not release assays, because that would substantially slow things down. And actually, so there's a link into another question that's in the chat, and Joris has mentioned this, and it's extremely important. It also came back to Martin's comments. So to tie all those pieces together, it's having really good analytical tools on the quality side are essential to characterize and then control these products. And I would argue that the control is not at the level of batch release. Those are confirmatory tests. It's all in the in-process controls along the way, and quality is built in from the beginning. So the final testing is important, but done by competent manufacturers under competent regulatory constraints. You know, is sufficient, I think, in this case especially. But to pull Martin into this is going back to flu. There is another question that Bruce Karpik mentions around good tools and SRID assays, etc. And Martin had mentioned in terms of elucidating these very complex interactions between specific antigens and platforms and clinical outcomes. I think having adequate and sophisticated, frankly, CMC understanding of what's going on, and as well as clinical understanding of what those different, how they translate is essential. And so a push to, I would push this not to regulators, but to manufacturers to update their methodologies to allow that better characterization, and particularly for the older platforms, like the egg-based platforms, to modernize that CMC, and I'll be very blunt, to move away from SRID assays and into better methodologies which are more applicable to broader platforms, including egg-based. And this is a push to manufacturers to again push regulators to update these approaches. I think is very good. I'd be very curious to see if Martin has a comment on that.
So, I mean, absolutely. One of the problems is if we're using an 80-year-old technology, which is egg-based vaccine production, we kind of are stuck with an 80-year-old quality control assay. And 80 years ago, SRID was about the best we could do, so we've remained with it. It's a huge drag on the process. I think all of the regulators here know that this adds weeks, if not months, to the process. We have to have the antibodies prepared, standardized to be able to do SRID. We have held several meetings to move away from this, but there has not been much progress. I fully agree with you. The manufacturers, we've got to encourage them, come up with assays that are better and propose these assays and show us the validation, because it's a really lousy assay.
Thank you, Martin. So let's stay up.
Joris,
yes.
Can we go to a bit more to the clinic and the question of the strains and the strain replacement, and the fact whether we can have a bivalent, a trivalent, or a tetravalent messenger RNA vaccine. I would like to formulate a question to Stefan and to Marco. What kind of data would you like to see if tomorrow a company would come up with a bivalent or a trivalent vaccine containing the old strain, but getting two other, by example, the Brazilian and the South African strain, into one vial? What kind of data would be needed to get such a thing to the market? And I think this is quite urgent to know what we are going to do, because there are data coming in that the South African strain seems to escape our vaccine. So this is quite urgent. What kind of data would we like to see?
Marco, I'm taking notes.
You want to start?
I can take a go, and of course I'm also wearing a semi-industry hat here as a consultant, of course. But I think it would be over the top to request a 5-digit clinical study like we just did for approval. On the other hand, we need to be reassured that a multivalent mRNA vaccine actually is able to raise relevant immunogenicity and antibodies and cellular immunity against all the valents, and not just one of them. So we will need, for me, at least a study in subjects, preferably subjects that may already have been vaccinated once with the classical strain, to see what will happen there in terms of immunogenicity. It needs to be fairly robust in terms of numbers of patients to take into account variability also in terms of age and comorbidities. But I think, coming back to the urgency, that it would be unreasonable to request a study in unvaccinated patients. Marco.
Yeah, no, well, of course, I agree with what you're saying, and we will not be requiring other clinical field efficacy study. That's pretty obvious. It's completely unfeasible. So it will be about immunobridging. How to do this immunobridging, that will have to be discussed, and much depends. Do we have a course of protection? Well, probably we won't have one, at least in the near future, if ever. So we will have to do an immunobridging without a course of protection, but taking into account a marker that might be suitable for doing this bridging. And of course, I believe that this should be done in a formal way. So we pre-specify criteria for success. And in principle, the bridging should be done to the primary series that demonstrated the clinical efficacy. So essentially showing the non-inferiority of the new vaccine after a primary series to the immunogenicity exerted by the original or the parent vaccine against the ancestral strain in terms of neutralizing antibodies titers. And of course, if you got like a monovalent variant vaccine, what we're looking for is that the neutralizing titers against the new variant strain will be non-inferior to the neutralizing activity of the parent vaccine against the ancestral strain. That will be the principle, and the same will be applied if it's multiple strain. And of course, if it's in this multi-strain vaccine, you're including the parent strain. Of course, you will have to demonstrate non-inferiority for that one as well. So that would be primarily how we will be looking at that. Of course, taking into account that when we say primary series, it means in subjects that were seronegative at baseline, which would be feasible for a while. And maybe there are also, archived samples from studies already done that can be used for that. Later on, when we move on, we will have to see how to do that. And of course, the other scenario is the one of the boosting approach. And then again, we will have to see how to do that and how to make sure that the booster response of the new vaccine, whether it's monovalent new variant or monovalent multivariants or multivariants plus ancestral strain, how to demonstrate that the immune response in terms of neutralizing antibody is non-inferior again to the primary series that demonstrated efficacy. This is in a nutshell how we would look at this. And I think we will be issuing a guidance document very soon. I hope in a matter of maybe a couple of weeks or even less, that will detail all this point. And I think something that we learn across the years that before issuing such guidances, it's good to touch base with other regulators in the world, and that's what we did, and that's why we try to agree with other agencies like FDA of Canada, but also discuss together with the ICMRA as a whole, whether everybody will be in principle happy with this approach. And I think it's important that we don't go out with guidances that say different things, because that will not help anybody.
Thank you, Marco. So there are two other questions. There is one question, and I don't know why I do not see my Q&A list anymore, but I remember the question from Olga. What about the transfer to other production sites of platform technologies? So what has to be complied with in order to transfer, and I can imagine to low and middle income countries, which is a possibility for these platform technologies. Who is Dean?
I can start yours. Many can jump in. Is the manufacturers in general, without sharing any specific secrets, have very good comparability approaches. So one is referred to as sort of a hub and spoke approach, where the core quality attributes are defined, and then additional facilities are pulled in within. These are all within the same quality control system, which is rather key, because then you have the same assays as applied, in theory, running the same way in different sites, and that has all those larger validation elements are coming down the line, but they are viewed as being acceptable at this point under the tremendous efforts the manufacturers are putting in. But in theory, that could be expanded to any other site with either the same or comparable quality management system. And some of these arrangements have been done, for example, with Serum Institute under AstraZeneca, but not under AstraZeneca as a separate piece. So those are, going forward. So in principle, this is it's a very complicated thing for a manufacturer to do, but they are successfully executing these very complex strategies to maintain comparability across sites. And in theory, that could be moved anywhere where there is a competent manufacturing facility in any part of the world with an appropriate quality control process. But maybe specific manufacturers want to comment as well.
Okay, thank you. Any other remarks?
No, I completely agree. And I would just add that reading the question right now, it looks like Olga would like to understand what is going to be, how the platform technologies principle can be leveraged in transferring the manufacturing sites. I would also ask the other way around. If we established a platform technology approach related to our manufacturing process, for example, and we consider that as a backbone to our platform, then to what extent we can manage different manufacturing sites and what flexibility we can have in introducing new manufacturing sites within the same platform principle. And I think, Dean, you already covered that basically. But I think that's the key point here, because the uncertainties related to any change to the manufacturing process, including additional new sites, are really what you need to handle when you want to approach a platform principle, right? You need to understand how to handle these uncertainties and how you can still define your platform even with these variables changing sometimes.
Thank you very much. So, yes, Dean.
Sorry to get off mute there. Joris, I want to loop back, just coming off of Ruben's commentary to an earlier discussion about different approaches, different platforms, and why not just put everything into the mRNA basket and go from there. And it's already been commented that's probably not a good idea, because we don't, as wonderful as the mRNA technology is being, has shown itself to be so far, we have other products coming along, and is a really key element of this. So first, to Bruce Karpik's point, we don't know how any particular technology will respond to a novel pathogen until we try it. And the mRNA vaccines have done well, and they may do extremely well in flu as well. And one would hope they do, because we certainly need better flu vaccines. But another element of this is, in addition to a platform being the best or a medium response in a particular case, and that's whether it's pre-fusion stabilized or not, these are all relevant issues. Across platforms, is that we are getting into a stage now where, at least in the UK, they are planning and will execute sort of the mixing and matching experiments with different platforms, which may yield some very important insights. So having multiple platforms, and therefore having the ability to look at different boosting options are extremely important, especially as we get into considerations about variants of constraint. And for the mRNA, although both our mRNA representatives here today were clear about this, is that while there may not be a molecular competition going on at an mRNA level, there is potentially still the issue of antigen competition, because those do have to be expressed, translated, and interact with immune systems. And all to Martin's points, while I'm, and the Health Canada group is very positive, as are Marco and Ralph and the FDA, of course, on the implementation of platform technologies to expedite review processes, as we've seen. There are differences between antigens, and flu, as Martin's pointed out, is a great way to look at that lens. So that doesn't mean we need to slow, it just means we need to be informed and collect that data. So I'll stop there.
Thank you. Can I move to a totally different question, which a lot of you will interest. So from Professor Robert Zauerwein, there are considerations and early initiatives to establish a human challenge model. What is the opinion about such data on the path towards regulatory approval for example, even multivalent vaccines? Who starts? I see Marco smiling.
I should be more serious, I know. Okay. No, I think we always been open to discuss human challenge studies, and even in the context of COVID-19, despite, of course, the question from an ethical perspective has been emerged, rightly so from the start, that, you know, without rescue therapy well established, it's not so easy to go through these kind of experiments. But overall, as also agreed by the WHO expert group, it's possible to identify a population in which you could run these studies, and you could minimize the risk for the participant. We always believe that this data could indeed provide additional evidence, particularly for vaccine for which it would not be possible to generate efficacy data in field studies, and for which the concept of immunobridging might be more complicated. You know, if you think about the fact that immunobridging might be something that we will be looking at in the future, but may have to be platform technology specific. So what to do if you got a brand new technology that has not demonstrated efficacy in the field, maybe with a different antigen? So that is a problem. And what to do, you know, then it will be extremely limited. And this is the reason why this kind of study could be rather informative and helpful. Exactly to what extent we could consider them pivotal for approval, I think it's too early to say. We will need to see what is coming up from the studies that we understand, at least in the UK, will be starting. And we will take it from there. But definitely is another tool that could generate evidence that could be important.
Thank you, Marco. Any other remarks on that? Dean? No?
Yeah, so no, I'll be happy to jump in. Yeah, I would echo everything that Marco has said. That there are certainly concerns, but it is potentially a very important tool in the toolbox, as Marco mentioned. And I think the fact that the UK studies are getting underway is very useful in that they will help define minimum effective dose, at least with the constructs they're using. That may have to be done again with different constructs, but it's a very good way to start in as safe a population as possible in those studies. And then once those pieces are established, then the possibility of doing those studies to look at different routes of administration with existing licensed products, nasal, et cetera, looking at different variant combinations. This is a potentially very valuable tool that has to be considered seriously and would be if it came to us.
Okay.
Can I just intervene a second? I fully agree with what Marco and Dean said, but the situation in the field is a little bit different. I know at least one center who is ready with the GMP production of the strain. They could do the trial tomorrow, but the ethics committee and the national authorities, they said no. They went to the WHO meeting, and the WHO meeting was rather negative. And so they said, no, we are not going to do this. Not in our country, full stop. So I think we need to open that discussion a little bit more on a higher level, that we get more support for this kind of trials if people want to do it, and if people get the right information. And we should not try to tell them that we shouldn't do this. We should leave it to the investigators and the volunteers to be pretty well informed and take their own decisions. So there might be a little bit of work for this on a regulatory side to say that these data could be important. Over.
Thank you, Peter. Peter, do we have time for a last question from?
Yeah, I think we can have the last question and then we can wrap up.
I don't know who Jean is, but I have a feeling who it might be somewhere in France. Could we know from RNA vaccine manufacturers amongst the data to be supplied to regulators post-marketing authorization? Were there many questions on manufacturing apart from shelf life study or mainly on the clinical part? Could they get most of the required CMC data prior to license or authorities accepted waiting for analytical data based on adequate risk management? Question marks.
Oh, it's $1 million question, isn't it? No, it's a good one. The discussion with the regulators, obviously for us, initiated as early as possible in general to try to define what was going to be a sufficient package for initial approval given the circumstances and everything. Whether we received more clinical or non-clinical or CMC questions, honestly, I'm not 100% sure. We have received different questions during the various regulatory procedures covering all aspects, and it's been a very intense exchange with the EMA, with MHRA, with the FDA, obviously, as usual, and in particular considering the timelines. We have squeezed review procedures of months in really a few weeks. So in general, I would say once we have agreed on the overall principles and requirements and the framework, it was relatively a straightforward conversation to put together what was going to be the required, absolutely mandatory requirement for the initial approvals, sometimes emergency use approvals, sometimes marketing authorizations, and what could be a post-marketing commitment or recommendations for further improvement. As I said in my presentation, obviously, with such short development timelines, it's normal that the post-approval phase includes a number of additional activities, further development commitments. This is something that I think everyone expected. It couldn't be otherwise. But in general, I would say that the CMC package, the overall quality package that in the end was provided to support the emergency use approval in the US or the conditional marketing authorization in Europe could be satisfactory compared to what we had agreed before with the regulators. And it was not really, I would say in general, it was not that different from what you would expect for a normally developed vaccine in general. With all the uncertainties related, of course, to the short development timelines, but I didn't see any shortcuts being taken besides squeezing the timelines, obviously. We need to generate additional data on the clinical side, on the non-clinical side to some extent, and on quality, obviously. But this is part of the normal regulatory processes. So I don't see a big difference compared to what you would normally do for any new product being approved in general.
Okay, thank you. Stefan?
I mean, as we are not yet in the full approval process, I cannot really comment yet on this, but would also agree that, of course, from the question, it's quite balanced, really also depending on the phase. And of course, I would assume there are, of course, a couple of commitments which in a normal approval would have been fulfilled already, but nevertheless, they have to be fulfilled. So the overall process is very similar. I guess the regulatory colleagues can comment better on that too, whether, yeah,
Marco,
they see it similarly.
No, yeah, I think, it's unavoidable for me to say that in the context of the conditional market authorization here, we're doing something that normally we would not do for conditional market authorization, okay? Because normally for conditional market authorization, the CMC package has to be complete. Here, because we are in an emergency, the law allows us to have an incomplete CMC package, and that's what happened here. And we believe we came to a conclusion on the benefit risk with sufficient amount of data also on the CMC, but unavoidably ended up having a number of post-approval commitment on manufacturing that normal would not happen for any vaccine approval. So for me, it's important to stress this. I think Of course, for us, that we don't have an emergency use authorization at European level, and we had to go through the conditional market authorization route, means that in principle, the standards also for manufacturing had to be quite high and higher than what you would assume or expect for an emergency use authorization. But we try to be as flexible as possible because we recognize the need to come up rapidly with a decision that will be in the best interest of public health. And that's why in the future, we will be refining how we approach, authorization in Europe, either conditional or whether we will have an emergency use authorization in the package of European tools in the legislation, which we are working on that. And we will see then what to do. But it's important to stress that indeed here, there were a bit more post-approval commitment on manufacturing than what we normally would see.
Thank you, Marco. Yes, Dean?
Yeah, I want to stress one key thing. I agree with everything that Marco said, but to leave people with a very clear idea is that this has been an ongoing debate, especially in the US, the emergency authorizations, in our case, an interim order, they are similar but different in some ways. And again, Europe has an array of different procedures as well. And as Marco said, they don't have an emergency procedure at a European level. But nonetheless, so the focus was on ensuring consistency of manufacturing. And no full validation is still not in place with most products, but it is very important. The standards around demonstrating consistency have not shifted. It's the evidence that would be normally submitted to support that. And there's a very important interplay that we know that the Europeans, the Americans, and ourselves all used in setting CMC specifications was the leverage against the clinical trial data. The dose ranging studies were critical in setting up appropriate quality management strategies for both the manufacturers and on the regulatory side. And then the demonstration of manufacturing consistency, even if these were not full CMC packages, as Marco refers to, were more than adequate to suggest that things were under control. And these are, again, under the stringent control of existing GMP systems and all these pieces leveraged together. So it was an incredible effort by the manufacturing side, but also, and I have to credit Marco and Marian Guber for working very closely together from an FDA perspective and ICMRA to pull this world quickly together as it was. And I think there's lots of lessons to be learned going forward. Over.
Thank you very much. I look at Peter now. I think it's time to conclude, isn't it, Peter?
Yep, I think so. Unfortunately, Melanie had to leave, yes, but she told me that Adam would say a few words together with you in order to finalize this, from my viewpoint, quite interesting webinar.
Adam first before I terminate.
Oh, thank you. And a really, really useful and helpful discussion, which we just had. And I think we've heard from a number of different people, which really reflects the learnings we've had over the course of the last year. As by necessity, we've had to come together and collaborate effectively, both regulators and industry and other parties. Some of those discussions are absolutely unprecedented, the speed at which things have moved. And I think all is driven by not cutting corners. It's about getting the right data and having the appropriate justification for that data to support the license applications. It's just wonderful that we have the legal mechanisms in place to provide some level of flexibility, but the regulators have absolutely taken the appropriate stance to make sure we've got products that meet the appropriate safety, quality, and efficacy standards. I'm very much looking forward to continuation of discussions. I really think that some of those processes for which we put in place to drive harmonization and to drive discussions, which in normal times would take an awful lot longer. I mean, some of the scientific advice procedures, you basically, when a company starts to work on a scientific advice, they're going to be it's going to be a good 6 months before they get that feedback, and it's just not going to work. And I really hope we can learn from the experiences we have. We can fully utilize the platform data that we generate and organizations generate, so that going forward we can do this again should the awful scenario occur whereby we have a further disease X, and we'll have to do even faster next time. Thank you.
Thank you, Adam. So Here, I would like to conclude by, first of all, thanking all the panelists for their input and their participation and their open speaking as we are used in IEBS. Also, all the presenters, and particularly thanking CEPI for this very nice collaboration. From what I heard, there are more question marks, and this is normal after this meeting than before. So a lot of things have to be discussed yet. And as you know, IABS is always eager to bring those people together where even extremely difficult, sensitive issues should be solved and should be discussed. So I think this is on the COVID side, and this can be an example for the future, where we all together can help, even where authorities can be happy to find themselves on a totally independent place, because as you know, IABS never takes position. We are there to forge consensus and for the sake of global public health. So that being said, thanks all of you. Particular thanks to the two people behind the scenes who made all this possible, Maddie and Abby, and particular thanks to Peter, who shortened his nights a little bit in order to make this happen, contact all the speakers, and organizing this in a very fantastic way. Thank you very much, Peter. And thanks all of you, and hope to see you soon in another occasion. So there will be a publication on the conclusions on this webinar, and this will be published in Biologicals if accepted as usual. And so I hope to meet all of you the soonest possible, and preferably in life. Thanks. Bye-bye.
Bye-bye.
Bye-bye.